Perfect. I have the screen perfectly. Thank you, guys. I see a lot of thumbs up, quite a bit. So, yeah, just a quick intro, right? We at deNOVO Biolabs, our mission has always been to accelerate diagnostic innovation and biologics development. And from the early beginnings in Bangalore to serving clients across the globe, our journey has been built on one core belief, right? That life science is about solving meaningful problems and just spreading awareness. And today's session is a reflection of that philosophy exactly. So, we'll explore the evolving role of ELISA in bioanalytical workflows in this webinar and the real world challenges that the teams face in modern method development and just practical solutions that have worked for deNOVO and the partners. So before we begin, let me quickly introduce our speakers. We have Dr. Dinesh Kumar, who is the co-founder and CSO of deNOVO Biolabs. Dr. Dinesh holds a PhD in life science from JSS Medical College, and he has over 18 years of experience in bioanalytical method development, validation, and regulatory alignment. He has also led the design and validation of immunoassays for biologics and biosimilars, and is continuing to bridge the gap between scientific innovation and business strategy. So, thank you for joining us, sir. And we also have Dr. Manjunath, who is the co-founder as well and the CTO of deNOVO Biolabs. Dr. Manjunath holds a PhD in biotech from JSS Medical College as well, and he brings deep industrial experience in antigen and antibody development, hybridoma technology, and protein purification. He's been instrumental in developing our affordable and automated purification systems, right? Protein purification systems, and has also co-authored a lot of patents, especially on monoclonal antibody innovation. Together, both of our co-founders bring decades of experience in translating R&D into reliable regulatory solutions. So, especially targeting diagnostics and biopharma. So, without further delay, let's dive in and explore the science, the challenges, the opportunities, and the next phase of bioanalytical method development using ELISA. So, again, just a quick reminder, guys, we'll have the floor open for Q&A. You can drop your questions anytime in the chat box. I'll be moderating those sessions, and feel free to ask any questions relevant to this topic, and our speakers will also address as many as questions they can. And yeah, I think I'll hand over the stage to Dr. Manjunath now. I think Dr. Manjunath is on mute. Just a reminder. Over to you, sir.
Yeah. So is it visible?
Yeah, we can have the screen, full screen now. Yeah, perfect. Continue, sir.
Yeah. Good afternoon to all the participants in this webinar. I welcome you all on behalf of deNOVO Biolabs. I thank you all for joining today's session on challenges and opportunities in bioanalytical method development by ELISA. In today's webinar, we'll explore how effective and precise reagents ensures quick and efficient ELISA method development, ensuring the sensitivity, reproducibility, regulatory compliance, and scalability for bioanalytical assays. I request the participants to note down the questions or write down the questions in the chat box, which can be answered and discussed at the end of the presentation. In this discussion today, we will explore both the technical and strategic aspects of ELISA-based bioanalytical method and how deNOVO Biolabs supports partners with comprehensive in-house expertise. We will move from broader context of role of ELISA in bioanalytical method development and discuss key challenges in method development and solutions to overcome them. Highlight the solutions which are provided by deNOVO Biolabs. The goal today is to blend scientific insights with practical applications. In the end, we will have an interactive session with the participants. deNOVO was founded in 2013 by a group of professionals with experience in science and management. Our R&D laboratory is located in Bangalore. We are one of the pioneers in indigenous development and characterization of recombinant proteins, monoclonal and polyclonal antibodies for bioanalytical assays, such as pharmacokinetic assays, immunogenicity, diagnostics, which are largely useful for preclinical, clinical studies, and quality control assays, which are used for the biological products and manufacturing of diagnostic grade ELISAs and rapid tests. The demand of developing biosimilars and new biologic drug is in traction from few decades now and will continue to be the potential domain of biopharma. This increasing demand of localized critical reagents is the opportunity for developing indigenous capabilities. We, as India's integrated antibody and ELISA solution provider, have been supporting the Indian and global players for their drug development projects by providing high quality, validated products and custom services. Our infrastructure is well equipped to execute research and development activities for antigens, antibody development, and further production of semi-finished and ready-to-use products. Our facility includes ISO 7 clean room for mammalian cell culture, microbiology, molecular biology, and production for R&D and manufacture of reagents and ready-to-use ELISA kits. In order to meet the regulatory requirement of biopharma industry, we maintain ISO 9001:2015 quality management systems, carry out all the animal studies under CPC SCA accreditation, and IBSC for our internal R&D activities. So, ELISA as a technique remains the cornerstone for bioanalytical testing from drug development to quality control, offering an unmatched reproducibility when designed and validated correctly. ELISA is one of the important techniques in biologics and biosimilar developments. Regulators like the FDA, EMA require validated ELISA methods for PK, PD, ADA, and HCP analysis. Despite new technologies being developed, ELISA remains the benchmark for reliability and reproducibility. However, ELISA's reliability depends on the quality of the reagents and validation methods which are used. Despite the ELISA strength, developers face multiple challenges that impact the method development and validation. ELISA challenges are multifactorial: cross-reactivity, matrix interferences, inconsistent reagents can distort the results and thereby fail the regulatory compliances. Moreover, translating manual ELISA setups into automated systems can change assay behavior if a robust method is not developed. Addressing these issues requires full reagent control and process understandings. These are precisely the areas where deNOVO adds value through integrated and in-house solutions. Each of these challenges inspired us to bring all the ELISA reagents development under one roof. So one of the biggest challenges in ELISA development is the antibody cross-reactivity. Cross-reactivity occurs when an antibody binds to unintended readings. Just a technical glitch. So cross-reactivity occurs when an antibody binds to unintended antigens that share structural similarity or epitopes with the target analyte. This leads to false positive or inflated readings, reducing the assay specificity and accuracy. One of the major causes of antibody cross-reactivity is the shared epitope structures, that is the homologous sequences from the related protein species or families, which may have overlapping amino acid sequences. Shared epitope structures can lead to cross-reactivity, reduced specificity in ELISA. So, what are the strategies to minimize this shared epitope cross-reactivity? We prefer to generate a monoclonal antibody or antibody pair and check its specificity against a single epitope. After we generate the antibodies against a specific protein, during antibody screening, we perform cross-reactivity panels tests and test the antibody reactivity against a library of related proteins, related isoforms of the proteins, or host proteins. We confirm the specificity of the antibodies by Western blot and make sure that the antibodies bind only to the target protein and not to cross-reactive protein samples. The affinity of the generated antibody plays a critical role in the sensitivity. Therefore, we test the antibody affinity to the target protein and cross-reactivity by using the SPR techniques. To give an example, when we want to generate an antibody, say against rituximab drug, which has a mouse CDR region and a human antibody backbone, we have to generate antibody specific only to the CDR region and check for having no cross-reactivity to its human backbone. So this will reduce the cross-reactivity in ELISA to human IgG when biological matrix like serum or plasma is used. So this strategy makes the antibody very much suitable for use in ELISA and ensuring minimal cross-reactivity and reliable detection. Next challenge is impurities in the reagents. The antibody specificity starts with the quality of the antigen used for generating the antibody. If the antigen contains impurities like the host cell proteins, aggregates, truncated fragments, the immune system of the host animals used for immunization, this will raise antibodies against those unintended components as well. This can cause serious cross-reactivity issues in ELISA, where antibodies start binding to the off-target proteins, producing false positive results, and increasing the non-specific background signals. Also, when the impure proteins are used as standard calibrator in ELISA, they may lead to false quantification, thereby giving inaccurate or inconsistent data. So how do we prevent or control this? So we have to verify that the purity of the proteins is greater than 95% before immunization into host animals. We have to test this antisera of the host to ensure that no pre-existing or cross-reactivity antibody is present in the host sera. To give an example, let's again consider the rituximab drug. When we want to generate an antibody to rituximab, we have to separate the CDR regions from the antibody by enzymatic digestion and purification. Then use the CDR fragment only to generate the antibodies and check for having no cross-reactivity to its human backbone. This will reduce the cross-reactivity in ELISA to human IgG when biological matrix like serum or plasma is used. Another aspect of cause of cross-reactivity in ELISA would be the use of polyclonal antibodies in ELISA, which recognize multiple epitopes. Polyclonal antibodies are a heterogeneous mix of immunoglobulins produced by different B cell clones in an immunized animal when a whole protein is immunized. So also when polyclonal antibodies are made, different animals produce slightly different antibody repertoires, even when immunized with the same antigen. When polyclonal antibodies are used in biological matrices, for example, serum, plasma, tissue lysates, the structural analogs or degraded protein fragments may mimic the target epitopes, leading to false positive or elevated background signals. Polyclonals cannot be reproduced identically once the host animal is gone. Therefore, maintaining long-term assay consistency and batch-to-batch variability can be very challenging. In ELISA, all these factors means multiple challenges like increased background noise, reduced assay specificity, and inconsistent data. So what are the strategies we use to mitigate this challenge if we need to use the polyclonal antibodies in ELISA? So the solution is to use antigen-coupled affinity columns to purify and isolate only the antibodies binding to the intended target proteins only. After the antibody purification, cross-adsorb by incubating the antibody preparations with potential cross-reactive proteins to remove the non-specific antibodies. Again, for example, let's consider the rituximab drug. So when we want to generate polyclonal antibodies to rituximab, purify the antibodies from CDR fragment immunized antisera. Deplete the antibody specific to the human antibody backbone. to get antibody specific to CDR regions only and having no cross-reactivity to its human backbone. Also, try to use the polyclonal antibodies in combination with monoclonal antibody, which would offer a balance between accuracy and the signal strength. After the challenge with the antibody, next challenge is the matrix interference, which refers to the impact of non-analyte components present in biological samples. For example, serum, plasma, urine, cell culture supernatant that may alter the ELISA development processes. In simple terms, the components in the sample interfere with antigen-antibody binding or signal generation, leading to an inaccurate analyte quantification. Matrix interference in ELISA happens because the biological matrices contain non-specific binding proteins like albumin, globulins, endogenous antibodies like rheumatoid factors, heterophilic antibodies, lipids, phospholipids, hemoglobin from hemolysis of serum, complement proteins, and proteases. Variable pH or ionic strength, all these components mask the epitopes or bind to the antibodies, which may suppress or increase the ELISA signals. To minimize these risks of matrix interference, we use blocking buffers with proteins and non-protein components to saturate the non-specific binding sites, or we can optimize the buffers by adjusting the pH, ionic strength, and concentration of detergents to reduce the non-specific interactions in the ELISA. We may also prepare the standard curves in the same matrix as these test samples. So, use of high affinity monoclonal antibodies provides a strong specific binding, which reduces susceptibility to interference from matrix proteins, thereby reducing the matrix interferences also. So the next challenge typically we face, that is the variability and reproducibility in ELISA, which depends on the consistency of reagents, primarily the antibodies, standards, substrates, and other buffers used in ELISA. Even minor changes in these components can cause significant differences in assay performance, signal intensity, and quantitation accuracy. This issue becomes more pronounced when reagents are sourced from multiple suppliers and change in suppliers or manufactured in separate lots. Reagent variability leads to poor assay reproducibility, calibration drift, and validation challenges. Because of this, the ELISA methods behave differently between labs or between users, which is a very cause of concern. To address this challenge, we have to minimize the reagent variability. Use a single source wherein manufacturing of critical reagents ensures complete control over formulation, purification, and QCs. What we do is we do lot-to-lot bridging. That is when new reagent lots are introduced, we perform parallel testing with the previous lot of reagents. We have in place a stringent quality control, wherein routine testing for purity, concentration, and binding performance is done. Whenever these samples are received for testing, it's always better to freeze these samples in smaller aliquots to minimize the freeze-thaw degradation. And the documentation and traceability is a top priority to register and maintain the reagent lot records, COAs, and performance trends, which is further useful for regulatory compliance, which is also a part of a challenge and point of our further discussions. Regulatory compliance in ELISA refers to ensuring the assay design, validation, execution, and documentation meets the international guidelines for bioanalytical and analytical method validation, which is set by the regulators. Since the ELISA data are often used to support product release, the stability, process validation, or say biosafety testing, the methods must always comply with the requirements set by the agencies like the FDA, EMA, and ICH. The table shown here mentions what compliances are required to be followed for the immunogenicity assays, the analytical and ligand binding assays like PK or pharmacokinetic assays. The compliance assures that the results from the ELISA method are accurate, reproducible, traceable, and scientifically defendable. We at deNOVO Biolabs developed and validated the ELISA methods to ensure performance of the assay to be robust and reliable. The ICH M10 guidelines is recommended for ligand binding assay validation to ensure the reproducible and robust data for regulatory decision making. When we take a pharmacokinetic assay as an example of ELISA method development, some of the validation parameters which needs to be followed are the specificity or cross-reactivity, which assesses the interference from related molecules and ensures the measurement of analyte without the interference from structural related compounds. The selectivity, which addresses the matrix interference assessments and needs to satisfy for the greater than 80% of the tested matrices. The calibration curve should be established with more than 6 calibration points in standard curve, and additional anchor points can be added to top or the bottom points to facilitate better curve fitting, and more than 75% of the calibration points should meet the accuracy within plus or minus of 20%, except at the LLOQ and ULOQ, which is plus or minus 25%. Acceptance criteria for accuracy and precision is 20%, except at the LLOQ and ULOQ, which is allowed to be 25%. The total error is calculated combining the accuracy and precision and allowed to be greater than 30% and 40% at LLOQ and ULOQ levels. The dilution linearity is performed above the ULOQ levels for QC points for the hook effect testing and should meet the standard acceptance criteria.
Miss Pravina, I think you're on unmute. I think Miss Pravina is on unmute. So, yeah, thank you. Continue, Dr.
Okay, sure. Yeah. The stability of the assay is to be performed by freeze thaw for short term and long term. This should follow the standard acceptance criteria for all the CC and QC points. Together, all these discussed criteria demonstrate that ELISA method is fit for purpose for pharmacokinetic assessment in non-clinical and clinical studies. Finally, the takeaway message is that regulatory compliance is what transforms a simple ELISA method into a reliable analytical tool that regulators can trust. Let me take you through some of the products developed by us after overcoming all these challenges discussed here and are ready to use for the clients. We have been providing these products for biosimilar research and quality control and batch release assays to domestic and global biopharma companies. We have recently included bioprocess monitoring ELISA kit for generic E. coli, HCP, and HCD with global standard of performance. We have developed a wide range of validated antigens and antibodies portfolio for vital diagnostic markers for use in the lateral flow assays in diagnostics. So deNOVO is well known for its supply of optimized assay reagents for ELISA, such as super sensitive TMB substrate, blocking and diluent buffers for ELISA method development and manufacturing activities. At deNOVO, we also provide variety of customized services to generate monoclonal antibodies in mice and polyclonal antibodies in rabbit, goat, guinea pigs. We also provide bulk antibody production services for the diagnostic test manufacturing, and we do have a vertical to provide custom lateral flow assay development. We also undertake services for antigen antibody binding kinetics or KD determination. ELISA-based bioanalytical method development and validation as per the regulatory guidelines and tech transfer to the customer site is also our USP, which is utilized by many of our clients. Molecular biology services for gene, probe, primer synthesis, recombinant protein expression, and purification are also available with us. This is a list of some of our clientele, which include global and domestic biopharma companies, vaccine companies, diagnostic manufacturers, and premier research institutions. So please do contact us for your specific requirements with our products or customized services, which we can offer at highest quality and competitive pricing. So I thank you all for your participation in this webinar. Please reach out to our team through the contact details provided here, or connect with us on LinkedIn or future updates and partnership opportunities. So let's take in the questions from the participants and discuss further.
Lovely.
Yeah, thank you. Thank you all.
Thank you, Dr. Manjunath. Yeah, I think the floor is now open for any Q&As. If you are comfortable coming on camera, we'd encourage that. Or if you want to drop your questions in the chat, please do so. The session was very informative. Thank you, Dr. Manjunath.
Thank you, Wes. I hope all the participants got an idea of ELISA challenges involved and solutions which they can use for their ELISA testings.
I hope so. I think we have a question from Rishika. Feel free, Dr. Dinesh or Dr. Manjunath, to take up. I think we have two questions right now. So Rishika asks, when purifying antibodies using antigen-coupled affinity columns and cross-absorption steps, how do you ensure that antibody yield remains high while maintaining specificity?
Oh, that's a good question. So generally what happens is during when using antigen coupled columns, affinity columns, the tendency is that the antibody gets diluted because the antibody specific to the antigen is very less in a polyclonal antisera. So what you can do best is purify the antibody first and then go for a concentration step wherein you can keep it at whatever concentrations is required by your purposes. And yeah, that is the one aspect you can do.
Cool, lovely, great answer. We have one more question from Kavita. She asks, are there any specific points we have to take care of while maintaining the reagent composition, especially based on the type of sample we use? For example, urine or blood sample?
While using urine or blood samples, generally you have to consider what dilution you have to use. So you have to optimize the buffer in your ELISA methods if you want to use a diluted sample, or if you have to use a neat sample, so you have to validate, or you have to develop your ELISA method and validate for using the neat samples there. So if you want to dilute it, use a buffer for dilution, and it has to be incorporated in ELISA method development steps and validation steps also.
Perfect. Thank you. We have one extra question from Devendra, who asks, how can competitive binding ELISA and Western blot be used to confirm antibody specificity?
In competitive binding, what happens is there is a competition created between your antigen and the antibody being tested. So only there what happens is the antibody specific to your antigen is picked up. And this also is done by Western blot, wherein when you load the protein samples, when you probe it with your antibodies should specifically detect a single protein and not any contaminant protein if they are present in your protein in the Western blot. So you can say if multiple bands are getting picked up by the same antibody, then it is not specific to your protein of interest, and there may be cross-reactivity with other proteins present in your protein sample.
Got it. And Gurudatta firstly appreciates our presentation. Thank you, Gurudatta. So he also asks, what are your thoughts and opinions on specificity and background reduction while working on detection of target proteins using ELISA for blood?
So Dr. Dinesh, do you want to take up this question?
Yeah, sure. It's a very interesting question here while addressing the specificity of any assay. And when it comes to blood, it's a complex matrix. So one need to perform the interference studies and what the dilution is optimum for the particular assay. And if the issue persists, then there are multiple approaches one can think of, like optimizing the ELISA with different blockers and diluents. And characterization of the antibody is very important here. The antibody specificity and affinity towards the target ligand. So I would say here to eliminate or to reduce the interference from the blood, serum or plasma matrix, one need to work out extensively on the blockers and diluents, and addition to the working on the antibodies.
I see. I think we have two more questions, Dr. Dinesh, before we kind of wrap this up. So Muruganandam asks, what is the typical percentage of antigen-specific antibody in polyclonal antibody pool?
Yes, this is a very challenging thing. See, when it comes to the high specificity of antibodies, we have to compromise on the yields of the antibody. So in general, the yields will be very less. I can say that maybe 5 to 10%. If I define in terms of percentage, we get target specific antibodies in that way. Yeah.
Lovely.
Next.
Thank you. I think Shailesh also wants to know some common challenges associated with saliva sample.
Sorry. Okay. What are the common challenges associated with saliva? See, each matrix is very unique for that matter, and each matrix has to be evaluated for the suitability in the assay. So one needs to do this assessment at very early phase of the assay development itself, working with the target matrix. It may be saliva, it may be serum, plasma, or the urine matrix. So early phase of development reagents, you need to qualify for the interference study that has to be done extensively with the different combinations of blockers and diluents, which are, some are commercially available, like protein-based, non-protein-based blockers and diluents are quite suitable for such interferences to reduce.
Perfect. I think we have a few more connects and contacts on the call. So, you guys feel free to ask any questions. We would definitely encourage your feedback. And I think we have a few interested partnerships already in the chat. So good to know and happy to see active participation. So any more questions, guys, feel free to, ask us. We'd be, happy to answer. And we have a question from Pravina, Dr. Dinesh.
Yes. Yeah, I could see that.
Yeah, so, she's asking if we could suggest her some tips while tagging antibodies for platform development.
Yes, the widely used antibody labeling is like biotin, HRP, ALP for the ELISA-based assays. And depending on the assay design and the application, we could suggest appropriate labeling of antibodies. And like to extend it to further, we do provide services for custom antibody labeling. And if you are okay to partner with complete assay development, which is something based on the ELISA, we can take up end-to-end service as well for right from the reagent development to hand over you ready to use method, which can be transferred to your laboratory. And we do hand holding right from beginning to until it works well in your own lab, and with in front of you, you can see the assays performing.
And also to add on to some of the tips what Pravina is asking, maybe you can consider that while biotin conjugation and all, you should not have interfering chemicals which can interfere with biotin, like trace present in the sample, or some amino acids, free amino acids present in the samples. You have your protein samples or antibody samples in a purest form of a buffer, free from any of the preservatives and contaminants.
Perfect. I think we are almost at the end of the call. Appreciate the questions, guys. Welcoming your feedback again. If there are any more questions before we get into the last stage of this lovely, insightful webinar, we would love to take them and just kind of nudge you guys in a direction where if it can help you even in the slightest way possible, then we have achieved an impact here on this call. Appreciate it, guys. Appreciate it for the kind words. I see a lot of messages of appreciation. So thank you, guys. Thank you so much. Yeah, I think cool. We have no further questions, and which reminds me to just take a moment and thank our lovely speakers, Dr. Dinesh and Dr. Manjunath, for such an incredibly insightful session, and the experience and the clarity you guys bring just makes this complex topic very accessible to our entire biotech community. And a big thank you as well to every participant who joined us today. We hope this session gave you practical takeaways that you can apply in your own work. And if you'd like to continue the conversation or explore deNOVO solutions for assay development or any other process, please visit our website or connect with us directly on LinkedIn. We have exciting stuff, very informative stuff happening on LinkedIn. And we'll also be sharing the recording and key highlights. So until next time, just stay curious, stay innovative, and thank you guys for joining us once again. So you have our emails. We'll also be sending a follow-up message after this call. So thank you, everyone. Thank you, guys. Appreciate it.
Yeah, thank you. Thank you for participating in this webinar. We see you soon again for the next webinar.
Take care, guys. Thank you.
Thank you all the participants for joining us today. And we take this opportunity of these interactions, and we hope to continue our conversation in some of the new partnerships to be begin. And I once again thank you, Vej, for coordinating and organizing this online webinar. Vej, I think we can wind up the call with this.
Yes, I think so.
If there are no any further questions or any conversation.
Sure, Dr. Dinesh. We can wind up the call. Thank you, all of you, for joining in.
Thank you. Thank you once again. I see Mr. Hari is on video. Okay, I think there is no more conversations, so I'm going to stop this. Thank you all. Thank you very much for joining us today, and see you in next webinar soon.